Hi guys, we're going to talk today about our fourth and final macromolecule that we're going to be talking about. And today's macromolecule is lipids. Lipids, you may not be familiar with that word, but I promise you're very familiar with some other categories of lipids, and that is the fats. Right? There's all this butter, and fats are what make these poor little mice look nice and chunky. He's got more lipids than this guy does. But that's not the only thing that lipids do. They are very important in cell signaling. They're very important in the structure of cells, and we're going to talk about that here in a minute. But hopefully by now you're pretty familiar with the common theme that they are all composed of carbon, hydrogen, and oxygen in a long hydrocarbon chain. In this category of macromolecule, we have fats, we have phospholipids, and hopefully by now you also know that phospholipids are in cell membranes. Oops. And then they also form steroids, and that's going to be very important in cell signaling when we get to cell. But lipids also tend to be the smallest of all of the macromolecules because they don't tend to form polymers. They are made of smaller subunits, excuse me, but they are not in a long continuing chain like all of the rest of the macromolecules have been up until this point. They have kind of a unique structure, and that structure is a tail and a head. That's what we call it. This is the glycerol head portion, this gray piece here. You see that that's a 3-carbon sugar with a bunch of OHs, and if you remember that OHs typically equaled ol or alcohol, all different kinds of alcohols. So glycerol is an alcohol. And the tails, these fatty acids, are all made of carbon and hydrogen, and with a oxygen here and an OH here that helps it bind to the glycerol. Now, fats and lipids and different, depending on which kind you're talking about, they can have 2, they can have 3, they can have 1 tail. And the tails can be short, they can be long, they can be straight, they can be kinky. So it just depends on which particular lipid you happen to be talking about. So this is a triacylglycerol. Triacylglycerol. Say it again. Triacylglycerol. Just got to practice. 3 fatty acids are linked to a glycerol. So we have 1 tail, 2 tails, 3 tails, and we still have this fatty acid or this glycerol head. And in between, they are linked together with what we call an ester linkage, which is a carbon connected to an oxygen. And they're put together with dehydration synthesis. So just like all of the other molecules are made with dehydration synthesis, so is fats. The only difference is they're not built combining um, similar monomers. Their monomers are a glycerol and a fatty acid. It's putting those two things together, but it is still releasing water and forming a bond that is called an ester linkage. Okay, so this is a fat molecule. Okay, triacylglycerol. And these long chains, do you think they are polar or non-polar? Now look at them. Look at carbon. Carbon, how many bonds does carbon want to form? It wants to form 4 bonds. Do these carbons have 4 bonds? They actually do. Or one's bond to another carbon, and then hydrogen's all around it. The ones in the center are bound to 2 carbons with hydrogen top and bottom, but they're all happy. So there's no partial negative or partial positive charge. So that means that these chains tend to be non-polar, very non-polar, in fact. And so what do we know about non-polar? Is it hydrophilic or hydrophobic? Well, we know that water is what? Water is polar. And since water is polar, it is sticky, and other things that can bind with it are going to be also polar. And we call those things hydrophilic. Hydrophilic means water lover, And then hydrophobic means I hate water. Okay, so nonpolar is hydrophobic, polar is hydrophilic. Their function, so they can have a lot of different
Energy.
functions. They can store energy, and they can store energy in a couple different forms, but it tends to be very, very rich storage, which is why there are some people that have a tendency to store fat. We call them survivalists because back in the caveman era, when food was few and far between, the people that could store fat as an energy source survived better than people who couldn't store fat as a good energy source. Um, because it tends to be very rich and it can store up to 2 times more energy than carbohydrates can store. It also fills in around your organs and cushions them. Some people too much, but that is an important function to insulate it. I mean, think about all the like hard bones and things that you have in there. You don't want um, those to be poked and prodded with by your bones. And then it's also very insulating. It insulates your body. Think about whale blubber, how insulating it is to that whale when they go into the deep, cold oceans. So that tends to be very insulating. It also tends to be waterproof. If you think of things like ducks, they have waterproof covering on their some of their feathers, and it is made of fat, and it's kind of repels water because it is hydrophobic. Anything that repels water is going to be waterproof and can form a waterproof coating. Okay, so now let's talk about saturated versus unsaturated. And you've probably heard this if you've talked to anybody about a diet or heard things on TV about diets. They say to stay away from saturated fats, and that's because of the structure of them. Remember how I said that the tails can be straight or kinky? Well, these saturated fats tend to have very, very straight tails. And that means that all the C's are bound to hydrogens. Okay. All carbons are bound to hydrogens on the top and the bottom. There are no carbon-carbon double bonds. And we'll see in a minute what that does. So what that means is that these tails can lay very, very straight up against each other. So if you have a or you have a glycerol head and you have fatty acid tails, they can sit really close together, and more of them can sit really close together. And the closer you get together, the more dense it is. And that means that it tends to be solid at room temperature. So you think of your butters and lard and Crisco, things that are solid at room temperature are made of saturated fats. Okay, this one is the one that clogs your arteries though, because again, it tends to be more solid more often than not. And so solid running through your veins, not a great idea. It gets caught on valves. And it gets stuck, and so contributes to cardiovascular disease. And these also tend to be more animal fats, so the things that you get from animals, things that you find in like red meats, dairy, things like that. Okay, now when they're packed close together, they tend to be solid, but sometimes they're not. Enter the unsaturated fat. Okay, look at what this has here. See how this one has a double bond right there? And that double bond changes the tail. It puts a kink in it. And so it makes it bend. And so if you have all these fatty acids and they're kind of, all over, they can't pack really close together because you have these fatty acid tails going out in different directions. And so they tend to be less dense, which means that it tends to be liquid at room temperature. Okay, so these things tend to be the oils and stuff from plants and fish. So the healthier fats like olive oil and avocado oil and stuff like that. So um, it tends to be more liquid. It goes through the veins more, better than saturated fats, so it doesn't tend to clog arteries as much as saturated fats do. Okay, so again, because of its structure, because of even one double bond, but it could be hundreds of double bonds depending on how big the molecule is, um, those double bonds, that little change in its structure is going to make it act completely different and make one thing kind of healthy for you and one thing pretty dangerous for you. So it's kind of an amazing thing that just a double bond can do that to the molecule. Ooh, that got stuck. Okay, so here is a trans fatty acid. And a trans means that it has a hydrogen on this side and this side, opposite. Okay, so trans usually means opposite. If you were to draw this as a cis fatty acid, those hydrogens would be on the same side. So cis, oops, cis equals same, and trans equals opposite. Okay, so again, that has to do with structure of the molecules. And again, we'll see how that plays a role in what it does, but it's just a different orientation. Kind of like how we were talking about those carbohydrates, where if you have a OH group pointing one direction, it's a carbohydrate or a starch that people can digest. And if it's pointing the other direction, then it's cellulose and it's something that we cannot digest. So it makes a big difference. So here are phospholipids. This is a specific category of lipids, and these are the ones that you find in every single cell membrane in your body. Okay, you have glycerol head, you have two fatty acid chains, okay, one, two, and then you have this unique phosphate structure right here in the middle that is negatively charged. Okay, now we're going to take a look here about how these different things affect. So let's look at here. All right, so we have this guy here that has a slightly negative charge because of that phosphate group. We have this part down, oops, let me do this real quick. You have this part down here that we know we already talked about is nonpolar because it has the carbons and they're all filled with hydrogen, they're all happy, there's no polarity there. So your fatty acid tails, these are hydrophobic. And the head, because of that polarity in that phosphate group, these are hydrophilic. Okay, so we say that they have a dual personality. So the way that they orient themselves in cell membranes is very, very important. And it all has to do with the function of its polarity, where it's polar and where it's not polar. Okay, so if we look at this here, okay, so that's the hydrophilic head. Those are the two tails of the phospholipid, okay, just drawn very simply. Um, if you were to just drop some of this in water, they would automatically form this little sac, okay? And all of the heads would face the outside, face the water, and all of the hydrophobic tails would face the inside because remember, they are repelled by water. They don't want to touch water. They're hydrophobic. So they would orient themselves to the inside so that they don't have to touch water. Okay. We also call this a liposome. Liposomes often like carry waste to the outside. And what you've noticed here is, what do you notice? You have layers of head here and layers of heads here. So what does that mean? Well, it means that now this thing, because both the inside and the outside can interact with water, that means I can carry things that are water soluble. Okay, lots of things in the human body are water soluble. But what that means is then I can carry stuff that is water soluble on the inside, and I can carry it to the cell membrane and carry it and release it to the outside. So they tend to be very, very helpful in terms of moving things um, around the cell, releasing it, bringing it in, things like that. So here is a picture of a cell membrane. Again, we have tails and tails facing each other because remember, they're hydrophobic and humans are full of water. So there's water on the inside, there's water on the outside. So they automatically orient themselves so that the hydrophilic, the water loving heads are facing the water, and the water-hating tails are in the center. And so every single one of your cells is surrounded by what we call the phospholipid bilayer. Okay, the it helps define what's inside and outside of the cell. Okay, so these are surrounding every single cell. It is the membrane of the cell. So phospholipids of the cell membrane. We call it a bilayer, a phospholipid bilayer, because it's two layers of phospholipids. Here's a picture of one where you can see the heads up here, the heads down here, and the tails in the center. And then what you have on going through it, these big purple things, those are proteins, and those proteins act as channels or pores because if you think about it, if you have that hydrophobic region in the center, how do things get to the inside? Okay, well, they can't unless they are going through a protected channel or being brought across in a liposome. So some things go right through, and the things that tend to go right through are very, very small and water soluble. The things that are bigger can't go through, have to go through these proteins. Okay, it basically this forms a nice barrier for the cell to prevent things from going in and coming out that the cell doesn't want it to. All right, so steroids. These tend to be your cholesterol. When you think of cholesterol and sex hormones, they tend to be ring structures, four fused rings together. Different steroids are created by attaching different functional groups to the rings. And they do so many different things. We're going to talk a little bit about them, but they have very wide and diverse functions. So here's just a couple of examples. We have cortisol. This is actually a stress hormone. It's kind of like goes hand in hand. Well, actually, both of these kind of go hand in hand with the fight or flight response. A lot of people, even just stress causes this. And so you have these different functional groups. So you can see how this region here is different, and then it creates a different function for the steroid. Here we have some sex hormones that are made primarily in the gonads. You have testosterone, estradiol, which is a female hormone, estrogen, but males and females both have it. Males and females both have testosterone as well. It's just the ratio that tends to be different in some individuals. And then you have progesterone, and that tends to tend to have a lot more of that when females are carrying a child. Progesterone goes up. That's actually what a pregnancy test tests for, is the progesterone level in your blood. All right, so this is an important cell component. Cholesterol is. Again, it's used in animal cell membranes. It is used to make other steroids. In high levels in the blood, though, can contribute to cardiovascular disease. So it's a fine line. You can't do without it, but you also can't have too much. So like a lot of things in life, you have to walk a fine line and make sure you have the right amount, because what ends up happening is that plaque builds up on the inside of your arteries and can eventually block the artery completely. Here's another good example of a cell membrane. These orange things are the glycerol heads, and then the black lines are the fatty acid tails. And you have cholesterol stuck in here in a couple of different places. And those cholesterol molecules are things that kind of help keep the cell membrane a little bit fluid and flexible. Because remember, the closer you pack lipids, the more solid they tend to become. Well, we need cell membranes to be fluid and flexible. So the cholesterol stuck in there helps the cell membrane to achieve that. And then finally, we have just a couple examples of the very different structures. So again, you can have one tail, you can have three tails, you can have two tails, you can have different additions to the heads, you can have lots of different. So there's a very big variety in lipids, and again, that translates to a big variety of functions. Okay, so that's it for lipids. And so what you guys are going to do now is you're going to do a matching activity where you're going to take a look at all four of the macromolecules and you're going to try to identify them based on their structure as lipids, proteins, carbohydrates, or lipids, proteins, nucleic acids. It's been a long day. So you're going to try to identify them and move them around. And then there's also in the PowerPoint is a some reading. Okay, some additional reading on the structure. So make sure you're reading everything that's in the slides because some of it you have to read the reading and then answer the questions. This is an assignment that normally would be done in class with cards and groups. But again, we are not allowed to do that right now. So I posted another discussion group below the PowerPoint. So please, I encourage you to talk to each other, to ask questions, things like that. So hopefully that helps, and I will see you soon.